Sure, we will. And this talk is now recorded. Yes, and Jeff, you have about 20 minutes, and then we have 10-minute question. Yeah.
Great. Okay, I'll go ahead and get started. Thanks to the organizers for inviting me to speak today. I'm excited to share some work that's been done at Johns Hopkins with the Gray Lab, as well as some work in collaboration with the Salesforce AI research team. The main topics are building language models for protein generation, as well as fitness prediction, and then how some of this has been applied to antibody sequences in particular. I'll start with a little bit of background on these protein language models. There have been two dominant paradigms for training these types of models. Typically, you'll assemble a large set of protein sequences. These can be evolutionarily diverse sequences or antibody sequences in particular. And then one of the first ways people trained these models was through a technique called masked language modeling. Here, the idea is to take an amino acid sequence, hide some of the residue positions from the model, and task it with predicting the identities of the residues you've hidden. This type of approach is useful for learning representations of protein sequences. You can imagine if you hide residues within an alpha helix, for example, the model needs to learn some sort of secondary structure representation to predict what residue was hidden. These models can also build up tertiary representations if you provide them with enough data. A few higher profile examples of this applied to fast evolutionary data sets are ESM from Facebook, as well as the ProBERT model. And then we've also trained a model called antibody, specifically for antibody sequences. And then there's a few other similar models to antibody out there as well. Another way you can approach self-supervision on protein sequences is through autoregressive modeling. Here, the idea is to provide the model with preceding residues and then have it predict what comes next. This can be useful for things like generating sequences, like I'll talk about later, as well as scoring protein sequences. In the antibody space, there's been a few applications of this. One that I think is interesting is this RNN LSTM model. Here, they trained on a set of sequences specific to a target, and then you show that you can generate more sequences against that target. For this talk, I'll focus at the beginning on a universal protein model. With a model like that, there's a few ways you can kind of tailor it towards specific protein design. One thing you could do is take your model and then train it further on a set of family-specific sequences. In doing so, you kind of limit the scope of the model to only generate for that family. Another thing you can do, if you have any annotations for your sequences, is sort of co-train a model with both the label for a sequence as well as the amino acid sequence itself. And then once you've done this, when you want to generate more sequences, you can simply provide the label, and it should generate what are called property-controlled sequences, where they align with the label. One model I'll talk about later is IgLM, which I've worked on, where we do this for antibodies, and we can tell the model to generate things like heavy chains, light chains from different species. And finally, a class of model that's been especially popular lately are these structure condition models. Here, the idea is to, rather than provide a property of the sequence, provide the structure that it folds into. And in this way, you can do structure condition generation and ideally come up with a sequence that matches the structure you provide the model. I'll talk mostly about two models in this talk. The first is ProGen2. Here, the general idea is to investigate how scaling up these autoregressive models impacts performance on protein generation as well as fitness prediction. And then I'll talk about IgLM, which is a specialized model for antibody sequence design. This is a controllable model, as I mentioned on the last slide, and we can use it to generate diverse antibody libraries. For ProGen2, we use two databases to produce data to train the model. These are BFD and UniRev90. These are very evolutionary diverse protein sequence databases. And we trained the ProGen2 family of models through this autoregressive task. We trained a range of models from 151 million parameters all the way up to 6.4 billion. As an initial check on the impacts of model scale, we calculate something called perplexity. I've shown the equation up at the top there. The general idea is to see for each position the model generates at, how many residues is it sort of deciding between. So a perplexity of 13 would mean the model is confused between about 13 residues at each step of generation. And you can see as the model scale increases from smaller to larger, the model becomes more certain in its prediction. So it's better fitting the data we train it on. After we've trained this model, one thing we can do is just generate completely unguided protein sequences. We did this for about 3,000 proteins. Here I'm showing the TM score for the predicted structure with AlphaFold versus the PDB. So this is kind of a similarity to proteins that have had their structures solved in their literature, and then the PLDDT, which is the confidence from AlphaFold. And you can see a correlation between the two. In general, models that look more like things that have been observed before are predicted more confidently by AlphaFold, but we do see some exceptions where a protein is confidently predicted, but doesn't look like things found in nature. Among these 3,000, we've picked out a few interesting examples. In most of these cases, the generated protein structure, which I have on the left in kind of the colorful representation, does look like something found in nature. For example, this top one is a solenoid protein. It resembles the tertiary structure of a natural family of proteins, but the sequence from ProGen that folds into this structure only shares 11% sequence identity with its closest natural counterpart. Sorry for the dog bark. We're not the first ones to observe this phenomenon. In fact, other researchers have found that when you train these large autoregressive models and generate sequences, you do typically find that they produce proteins that fold up like natural proteins, but do so with a pretty different sequence signature. Another thing we can do that I mentioned before is fine-tune this model for specific families. Here we've taken one of the ProGen models and trained it on a set of proteins that have this two-layer sandwich architecture. And after doing this, we can generate sequences that resemble this family. Here are just a few examples. This protein family typically binds a ligand. In these examples, I'm showing the natural protein that's kind of grayish outline, and the generated proteins with the structures predicted by AlphaFold shown in the colorful representation. And you can see that the natural ligand actually fits fairly nicely into a pocket held open by the generated proteins. So the model picks up on not only the sequence and structural characteristics of the family, but it also leaves some room for the protein to carry out what would have been its natural function. When we're generating, you can also enforce some control over the sequences you get from the model through a couple sampling parameters. One of those parameters is the sampling temperature. The other is nucleus sampling probability. The way we apply these to the model generation process is basically to readjust the distribution at each step of sampling. This equation describes how we go from model outputs to a probability of each residue. So what we're doing with temperature is basically raising the temperature to flatten the distribution out. So something that might have been 90% likely at a position, if you raise the temperature, it might become 60% likely. With nucleus sampling probability, what we're doing is truncating the distribution. So we can say that for each position, we only want to sample the residues that make up the highest 50% of the probability distribution. So this sort of restricts diversity as you lower the p-value. In these plots, I'm showing the effects of both further fine-tuning from one epoch to two epochs over the fine-tuning data, as well as varying these sampling parameters. You see mostly what you would expect. If you train the model further, both sequence identity in the left plot and with respect to the temperature shifts to the right, meaning they look more like the training data. And then if you look sort of within these plots, you can see as we raise the temperature, the lines shift left, meaning that if you raise the temperature, you get things that look less like those produced by nature. And likewise with the p-value. We also do this analysis looking at the structures, and it's the same takeaway. As you increase the sampling diversity, you get things that look less like those produced by nature. Now I'll talk a little bit about evaluating or estimating protein fitness with the ProGen models. Here we want to use a nice characteristic of the model training process to see if it correlates with fitness values of proteins. So as we're training the model, what we're really asking it to do is to predict conditionally the likelihood of a residue at position I given the preceding residues. This means that at inference time, if you have a protein sequence, you can sort of calculate the joint probability over the entire sequence, which should measure something like an evolutionary likelihood of the sequence. And then we want to see if this correlates with fitness values that have been collected in the literature. We've sorted our fitness values in the literature into two rough categories. The first are what we call narrow fitness landscapes. These are cases where the variants are just a few mutations away from wild type. So this is things like deep mutational scans and other point mutations. This plot is showing a comparison of fitness estimations from the ProGens models on the right in the blue versus some models from literature. I've highlighted just a couple from literature. One is Eve and another is Transception. These are models that take in the multiple sequence alignment for the family and use that information to generate fitness predictions. You can see that the ProGen models, they match the performance of these just from pre-training on large sets of evolutionarily diverse sequences. Something interesting in this plot, though, is as you scale the ProGen model size from the smaller one up to 6.4 billion parameters, we actually peak in performance in this base model, which only has about 700 million parameters. This is a bit surprising, and people in the literature have found different scaling behaviors of these models. But a big takeaway of this project was that scaling isn't always going to give you better performance. In fact, for these narrow landscapes, it might degrade your performance. However, when we look at broader fitness landscapes, these are cases where variants might be dozens of mutations away from the wild type or have highly epistatic landscapes. Here we do see benefits from scaling the model. We only have 4 targets here, which I've described on the right. But in most cases we see as you increase the model up from 100 or so million parameters up to 6 billion, you typically see consistent improvements in this fitness prediction task. One case that's especially interesting is this one on the far right. This GB1 protein has binding fitness data. And you can see that as we increase the model scale, you go from effectively no ability to discriminate binders from non-binders up to a much higher ability to do this fitness prediction once you get to 6.4 billion parameters. This target, just to give a little bit more insight into what we're predicting here, has nearly exhaustive sample or evaluation of possible residues at 4 positions here that I've shown in blue. This landscape is also highly epistatic, meaning that these mutations really aren't additive. So the model has to pick up on something deeper about the coordination of these residues that appears to only emerge when you scale the model into the billions of parameters. Coming back to antibodies a little bit, we've also trained a similar model on the observed antibody space database. Here we've taken this data set, clustered it down a little bit just to remove redundancy within the sequences. After clustering this down, we have about 500 million sequences. We've used this to train a model called ProGen2 OAS that has 764 million parameters. As far as I know, this is the largest model trained on OAS data. With this model, we can do some of the stuff I mentioned before. We can generate antibody sequences. Here I've generated sequences, folded them with our IgFold software, and then predicted aggregation scores for these. We find, in this case, as you vary these sampling parameters that I mentioned before, if you generate more diverse sequences, meaning higher T and P, you typically get reduced aggregation propensity. We think this might be useful for people building libraries with these types of models, as you would like your library to have lower aggregation risk. So maybe sampling with higher diversity can give you those properties. We did something similar for solubility as well. Here, we don't need a structure. We can just pass the sequence directly to the ChemSol tool. And again, we see that when you sample with more diversity, you get sequences that are likely to be more soluble. The last thing we did with this model was test its fitness prediction capabilities. Here we've assembled 2 data sets specific to antibodies. The first is kind of a binding fitness data set, and the second is a general fitness data set, which is composed of relative expression of antibodies as well as thermal stabilities. You can see in both cases, the ProGen2 OAS model that I have in green actually underperforms the models trained on evolutionarily diverse protein sequences. This is a bit surprising. We expected, and I think a lot of the literature expects, that a model trained on specifically antibody sequences should be better at these tasks because it's seen more antibody data. But we think that the real consideration here is, what are the evolutionary pressures on the sequences you're training on? So with antibody sequences, you really don't see the broader landscape of what mutations are allowed and what might lead to better expression or thermal stability. Whereas in these broader landscapes, you see all sorts of mutations. I do think that if we had a data set that was something like immunogenicity or humanization, then you would see advantages with the model trained on OAS. Next, I'll go a little bit into our model for antibody library generation. To give a little bit of motivation for this model, in antibody discovery campaigns, you'll typically start with some sequence library. These can be generated through random mutagenesis, error from PCR, and the like, to produce some diversity. Then they're typically screened over an antigen to find specific antibodies and adapted to some format of interest. So what we'd like to do here is replace this initial library randomization process with a generative model. The model framework is similar to the ProGen models that I mentioned before, except we add this controllable element. So here we're training our model with tags that indicate whether we'd like the model to generate a sequence from a specific species, such as humans, mice, etc., as well as the chain type we'd like it to generate. So in this case, if you provide the model with human heavy, it should give you a sequence that looks like a human heavy chain antibody sequence. Then we can predict these conditionally to generate libraries. Just like before, we can kind of provide the model with these tags and let it generate at will. If we do this, we sort of see a reflection of the training data in the sequences we get. In OAS, a lot of the sequences are missing residues at the end termini. So when you just generate with the tags without any other precautions, you'll get these incomplete sequences. We can overcome this simply by providing the first few residues. So this sort of primes the model to produce a full antibody sequence. And then we can do the same with other species in our training data set. So here I've provided the camel species tag as well as heavy chain and a few residues, and we get out what looks like a nanobody from a camel. The unique feature of this model is this infilling task we trained for. Here what we're doing is replacing a subset of the residues with this special span token. So what this is saying is at this position, there should be a set of residues. Maybe this corresponds to a CDR H3 loop, but we're just going to tell you something goes here, but not how many residues or what they are. And then during training, we put that span token in a random spot and move the residues that correspond to that region of sequence to the end. So in this way, the model can look at the sequence that becomes before and after your span, and then predict the missing residues in the context of those other sequence bits. And we can use this to do things like redesign CDR H3 loops conditioned on the preceding and following context of the framework. These are some results that we've gotten by applying this approach to a set of 49 therapeutic antibodies. Here I'm regenerating the CDR H3 loops. There's a few thousand sequences generated per parent antibody. As you can see, we get a range of CDR H3 loop lengths for different parent antibodies, meaning the model isn't just kind of agnostically spitting out sequences for any heavy chain. It's really tailoring these to the specifics of the surrounding context. So some frameworks the model thinks are more amenable to long loops, some for shorter loops. We've also investigated a bit the effects of these sampling parameters on generation. One thing I expected going into this is that as you tune these temperature and nucleus sampling probability values, you might get longer loops with more diversity. In fact, we don't really see any effects on loop length as you turn up the temperature or allow more unconstrained sampling. We kind of see this flat trend. So the model always predicts loops of similar lengths. What we do see diversity is in the content of those loops. So as we increase the sampling temperature and the nucleus sampling probability, you get more and more diverse sequences. Here we're measuring for the generated loops, how similar are they to the closest the closest neighbor within that set of generated sequences. And then as kind of a sanity check, I've also measured within the generations, how similar are they to the parent antibody? You can see they typically don't come that close to the loop you deleted from your starting antibody, which you wouldn't expect given that these are just supposed to look like CDR H3 loops given the framework. I'd like to wrap up just by and our funding that I've shown below. And I'm happy to take any questions.
Thanks, Jeff. Very much on time. I like a lot. Do we have any questions from the audience? So one question that I always have is when you have this huge language model of antibody sequences, 550 million or something. Mm-hmm. What if you trained again another model on another 550? Would they be the same, or would you have still undersampled the antibody space? And are you mixing together all different isotypes? And did you guys ever test whether there is an isotype bias? If you just made an IgM model, would it be worse at predicting sequence changes in IgG? Something like that.
Yeah, those are good questions. To the first question about if we trained a new model on the same data, would the results be different? I think one way to ask or to answer this question is to think about how well the model is capturing the data. For the IgLM model, we haven't calculated the perplexity value over a held out set, but for ProGen, we see that as you increase the model size, it better and better captures the data. So I think if you trained more models or bigger models on the same OAS data, you should better capture it, and then you'll get generations that look more like the training data as well as better likelihoods. The value of the likelihoods, of course, depending on what you're trying to use it for, as we show. To the second question, kind of about the data distribution, I haven't investigated too much what the biases in the OAS data are. I think I'm sure somebody here would know better than me, but I think it's mostly IgG and IgM, but I'm really not sure. I think it would be interesting to fine tune specifically for different isotypes if that has value either in understanding immune repertoire dynamics or building sort of tailored antibody sequence libraries, but we haven't looked too much into it.
And something crazy, if you were to train on mouse data and then try to optimize human data, would that work at all? Did you ever try something like that, or no, like cross-species stuff?
Yeah, that's an interesting question.
Shouldn't really, maybe some underlying principles are learned. I don't know. Yeah.
Yeah, there's an interesting paper where they train a model similar to the masked residue prediction models I mentioned for human antibodies. And there they apply this model trained on human antibodies to mouse data, and they show that if you take the changes from that masked residue prediction task that the model is most confident in, you can humanize mouse antibodies. I haven't seen anybody trained specifically on mouse and kind of try to learn an explicit mapping between mouse and human. It might be challenging to assemble a training task that does that, because I think you need some correspondence between the two. Maybe a sequence alignment of mouse and human data would be enough, but yeah, I think there's a lot of potential for those types of projects with data.
Yeah, and just for the record, I should say, since we're in the iReceptor seminar, so I think the iReceptor database is larger than the OAS database, and people always train on OAS data, but people should actually train on iReceptor data because it's not. So just for all the people in this room that are thinking about something similar. And,
yeah.
And then there is a question in the comment section, in the chat section. Do you see it? So
yes.
Yeah.
Yes. So is the training library available from the GitHub sites? I think the sequences for the antibody data from OAS, I have them online somewhere. I can try to link to it from the GitHub, from the IgLM GitHub. The training code for the models, I'm not sure about. I know for IgLM, it's very tailored to our compute architecture, but we use an open source framework called Hugging Face to train the models for both of these cases. So it should be really straightforward to adapt the model now that we've built the architecture to a new training setup. The data would be harder to reproduce, so we provided that.
Are there any more questions for Jeff? Then I have more questions. So I know that in some of the Jeff Gray papers, you guys are also talking or thinking a bit about interpretability. Is this something that you are also working on, especially with protein language models? What do they learn? You know, what can we actually learn biologically? I mean, it's nice that they can predict things or, you know, help us, but what do we actually learn biologically by using them?
Yeah, I think when we think about interpretability, we want to be careful that we aren't making too many assumptions in terms of the immunological and biological background. I have one project where we've looked at trying to take an antibody repertoire from an individual or a set of individuals that have responded to the same antigen and see if you can kind of untangle which residues in the antibody are coordinating that response. That's the project kind of is coupled with the anti-protein model that I mentioned. I think there's also been work using the attention weights from these types of models to try to understand something about antibody sequences. I think it's when you build these very large models, it can be hard to know exactly what are patterns that are significant or what are patterns that will eventually emerge if you add enough parameters. So it can be a little bit tricky, but I think asking the right questions that you have the data to ask is something we're interested in. We'd be happy to talk with anybody that has such data or such questions.
So what do you think are the biggest challenges in this field of applying language models to biological sequence data, or what would be the next steps of the field?
Yeah, I think something that I learned from the ProGen project is that paying attention to the data distribution is really important. We had trained the IgLM model and the BERT model prior to training the ProGen one and comparing what happens when you try to make fitness estimations for antibodies between specialized models and general models. And it kind of disagreed with my assumptions going in. So I think that thinking more carefully about what immune sequence data tells you when you apply these pre-training tasks to the data. I think further understanding what we're really getting from the antibody sequence data is going to be a big area of research going forward. My assumption now is that you can think of a model trained on the immune data as sort of measuring a likelihood of a sequence being produced by the immune system, kind of analogous to the general models predicting a likelihood that the sequence was produced by evolution. So I think under that framework, they could be really useful for things like screening antibodies for humanness and lower immunogenic risk. But beyond that, I think it's an open area of research what they can really be used for.
And maybe my last question towards that point, towards the trustability of them or something like that. So we know that there is a lot of bias going into generating these huge repertoire data. You know, this lab uses this PCR method, and the sequencing. Has anybody looked at how technical bias feeds into what comes out of it, or
Yeah, that's a really great question. I had at one point wanted to sort of address that by taking all of these different studies and categorizing them by the sequencing technique, and then see if you train, for example, on something from 10x, how does the model trained on that data perform, maybe by perplexity on data sets collected by some other technique, and see really, are these within the same distribution, or is there something shifting? We haven't had an opportunity to do that yet, but I think that could be one way to get at the biases in the data distribution.
That would be really nice, I think. That would be really nice. If there are no further questions, I don't see any, then we thank you again for talking here today, Jeff. Really nice talk. And the next seminar will be on October 27th, and I hope I will see many of you there again. Thanks, everyone. Bye-bye. Bye-bye. Bye-bye.